You know, this external. Okay, so do you see the old test? Okay. Do you see? Let's look at number 4. Okay, that's kind of similar to what we're doing this semester, or we've done this semester. Is label the following diagram with the specific regions, right? Okay. So how would I do this? Well, this is option 4. We have this entire long gene, right? Okay, and on the left we have an ATG, and below it we have a TAC, so that's our what? Start codon, correct. And we have a line that says gene, and then we have a stop codon here. Or really a series of stop codons, to be truthful. Okay, so we're going to go down the list, and to answer your question, you said, how do you do this? Make sure you address every single point. We look through this and you see a question, make sure you address that point. Another question, make sure you address that point. Another suggestion is to draw pictures. Pictures are worth 1,000 words. If I see a picture and you might not fully explain it, I might think, okay, you got the concept. So I do like pictures. I don't require them, but they do help. So let's get back to this example. So first one, approximate location of the promoters. Where would the promoters be here on this diagram? On the left or on the right? On the left. Okay. Would the promoters be here or would they be down here? Not closer. What do the promoters do? They recruit RNA polymerase, right? So they would really be down here. So our promoters would be down here. And we're going to see why in a second. And what were our 2 promoters? The 2 specific regions we identified? We have some numbers here. It says minus number, or pound sign, and minus pound sign. What were those 2 numbers? 35 and 10. Where is what is this going to be 35 or is this going to be 10 or is this going to be 10 and it's going to be 35?
35 and 10.
Okay, so minus 35 and minus 10 promoters. And what I'm trying to show, this is a strand of DNA. Okay, so we have our 35 region or minus 10 region. Okay, very good. Now, what are we counting down to? What is 0? The start. What is 0 here? So minus 35, minus 10, 8, 7, 6, 5, 4, 3, 2, 1, 0. What is 0? Start off. I'm sorry?
The start of the polymerase?
So polymerase transcribing. So yes, when the polymerase first puts off that first base of mRNA, start of transcription. So 0, and then we would under here identify start of transcription. All right. Now we're going down the list. We can now mark off number 1. We can now mark off number 2. What's going to come between the start of transcription? So here, RNA is being made, right? So you might even want to draw a little strand of mRNA. Okay, so where does the ribosome bind to? Where is the Shine-Delgarno sequence? So would the Shine-Delgarno sequence be here, or would it be here?
It's before it starts.
Before it starts. Here's a piece of mRNA. What's that Shine-Delgarno sequence? What's the ribosome binding site going to do for us? Bind our ribosome. So we're going to have it here. This is where our RBS, our ribosome binding site, is located. Okay, we go down the list. We've now shown the location of the ribosome binding site. Where is the start of translation?
After the stop codon?
Not after the stop codon.
It's the same time.
It's not, well, what you said stop codon. You're not stop, but the start codon. The start codon is where what happens? Translation, exactly. So this is the start of translation. I'll make a nicer diagram here when we're done. Okay, so we check off ribosome binding site. We checked off start site of translation. Where is the end of transcription and where is the end of translation? Which is going to come first, the end of transcription or the end of translation? Now, transcription is going to come first.
Translation.
Translation. Remember, mRNA had a leader and a lagger? We have a region before and after it, so we have to make more RNA on both ends, right? So our stop of translation comes first. And then our stop of transcription. How about I make a prettier diagram? Will that work? Okay. So we have our DNA. Okay. We have our ATG, PAC. We have our stop codon, or series of stop codons. What comes first? You guys tell me. I'm going to go left to right. What comes first? Promoter. Which number?
-35.
Negative 35. Correct. And what's next?
-10.
Negative 10. Okay. So those were some really conserved sequences. What's next? And what does the promoter do again?
It allows it to bind to it.
Exactly. The RNA polymerase is binding here, right? So the RNA polymerase can now go down and make RNA. All right. Let's go ahead and just draw the RNA that. I'm sorry. So let's keep on going. Where is the start of transcription?
0.
0. So right here is our start of transcription at our 0 point. So RNA polymerase comes and binds to these promoters. All right, sits on there, and it starts making RNA. So here we're making RNA. Now, the stop codon is right here. Are we going to stop making RNA at the stop codon? No, we're going to make a lagger. We're going to make a short sequence afterwards. Okay? So we'll mark that to say the stop codon there. We make a little bit more. All right. Where are we going to have our ribosome binding site, also known as a Shine-Delgerno sequence?
Right before the start.
Right before the start, but after the
0.
correct. The promoter, so the ribosome is going to have to assemble here. So our ribosome assembles there. So this is our ribosome binding site, also known as a Shine-Delgerno. Okay. Start codon, this is the start of what?
Translation.
Start of translation, making protein. All right, now we have our, at the start of translation, I'm going to use purple for protein. That's P. Okay, good. All right, our protein is being made. Where are we going to stop making our protein?
Stop.
At the stop codon, correct. Okay, so this is our protein, and this is our mRNA. All right, so now we're at the stop codon. Great. Where is the end of translation? The stop codon. All right, where is the end of transcription?
After that.
After that, exactly. End of transcription, end of the mRNA. End of translation, end of the protein. Okay. Someone said, how do you answer these questions? Put as much detail as possible. If I have a question thing, what I do, and that's how I do these test questions, I will have a rubric. And each bullet point is a sum point value. Like this question right here, if I recall correctly, each of these points was 2 points. And I look at the answer, and if it says that answer, good, 2 points. If it doesn't, 2 points off. And, you know, if it's kind of, you know, I give partial credit sometimes also, but pretty much if I have a prompt, make sure you address that prompt. There are some essay questions here. No, there's another page. If you look on part 1, it's 5 points each, choose 2. Compare and contrast bacteria versus eukaryotic systems with their RNA. We've talked about that one. Compare and contrast eukaryotic RNA and prokaryotic RNA. How to answer that question? Best way to do that would be bullet pointed. Okay, what's one difference? One difference.
Cell wall.
Not cell wall, no. Okay, so RNA.
Oh,
RNA. What's one difference between bacterial and eukaryotic RNA? Start codon, but a little bit more specific. You're correct.
Eukaryotic, it's always mostly AUG.
Mm-hmm. And bacteria, it's what? It's not always AUG. Yeah, about 10-20% of the time, or 10, it varies in the species, but it's going to be less than that. Okay, you're correct. Now what's another one? So we got one. Good. And how would I answer that? I would say 1, start codon. And then, as you guys were saying, bacteria, eukaryotic. Okay, what's another one?
The introns and exons.
Introns and exons, exactly. Very good. So we got introns and exons. Bacteria, eukaryotic. What are bacteria? Do they have them or not? No, they do not. How about bacteria? I mean, eukaryotic systems. Yes, they do. All right, another difference.
The times of translation, transcription.
Correct. Translation, transcription. All right, you'd say bacteria and eukaryotic. So bacteria would be what?
Same time.
Same time, right? As that RNA is being made and coming off, we've got our ribosome sitting on it making protein. How about eukaryotic systems? Different times. All right, one more difference. Actually, one more major difference.
Number of polypeptides.
Number of polypeptides. Okay, actually, we've got another difference after this. Number of polypeptides. Eukaryotic, 1 polypeptide per mRNA. Bacteria, 1 or multiple polypeptides. Remember we talked about operons? We can, and I want to emphasize, not all genes are operons in eukaryotic, prokaryotic bacteria, but they can be there. So we can have multiple or just 1. All right, and then one last difference. Processing. Exactly. Bacteria are going to be what? No processing, right? Eukaryotic systems are going to be processing. 5' colon seal cat, poly A tail. You can even throw in the introns, exons in there because you're pulling out the introns. So that would be the thing. Make sure if I say something, a nice bullet-pointed list is really, really a good thing. Describe row-dependent and independent termination. So what happens with row-dependent? And what are we terminating here? Before we start this, what are we terminating? I guess that'd be the first good question we want to ask here. What are we stopping? What are we stopping? What are we trying to terminate? Are we trying to terminate translation or transcription? Transcription. So what are we trying to terminate the function of? Of what enzyme? Polymerase. Which one? No, you're thinking DNA. If we're doing transcription, we're making RNA. So what polymerase? No? RNA polymerase. RNA polymerase. Now, okay, helicase, that's in DNA replication. That's in the helicase, that's the one that separates the genes. All right, neither here nor there. Row-dependent and row-independent. We're stopping RNA transcription. We're trying to stop RNA polymerase. Row-dependent. We have our RNA polymerase on there. And row dependent, we had that row protein that sits under RNA and moves up. And as it moves up, it's actually using ATP to do this. It pops off our RNA polymerase. Row independent, we had that what? That stem loop structure, right? Where it formed it hydrogen bonded to itself. And as it hydrogen bonded to itself, it pulled back on the mRNA transcript, and that's what pulled off the RNA polymerase.
Can you repeat that?
Okay. With RNA row independent, a stem loop structure forms. If you look at the sequence in this region, it's going to be what we're calling an inverted repeat. So here it would be, for example, A, G, A, U. And how the sequence is made is that when it's in close association, it will want a hydrogen bond to itself. Okay, and that hydrogen bonding will make what we call a stem loop structure. It's going to physically pull to itself. And that binding is going to actually, it's like a rope being pulled back. But yeah, I cannot, it's hard for me to emphasize the importance of drawing the pictures out. It's really, really good to draw pictures out for me. I'm a real visual person. Okay. All right. This is row. Okay, this is self-termination. So self-termination, we're going to have a stem loop structure form. Okay, that's self-termination or row independent. Okay. So you see what's happening? The RNA is looping back on itself. You see that? Okay, why is it doing that? It's because there's complementary pairing happening between that strand. You know, it's right here, that is binding to itself. And by doing that, do you see the purple thing right there? That is the RNA polymerase. See that, the orange thing right there? That's the RNA polymerase. You kind of see what's happening? As that stem loop structure forms, it's physically pulling the RNA that's being made out. And that will help to disassociate the RNA polymerase.
So row independent is synonymous
with self-termination. Correct. It's independent of the row protein. Row dependent has the row protein. So with row dependent, we do that before, the pink one right here, that's going to be the row protein. It's going to hydrolyze ATP, and it's going to move down our transcript. And as it moves down, it will physically kick off our RNA polymerase. Okay, so you see it's coming down here. It's physically moving up, and as it goes to that area, in the strict term of the word, it physically kicks it off. It pops it off. Okay? All right. Another question. Let's see here. Let's go back to that those possible test questions.
Are you broadcasting?
I am. I'm recording right now. I'll take pictures of this also. Okay. Here's a good one. Draw the entire, and this is a good one because it's going to give us a lot of, little points that we want to make sure we do. Draw the entire DNA replication fork with the specific enzymes used at each point. Be sure to identify both strands in this. Also, how does bacteria deal with excessive supercoiling and stress from DNA replication and the replication fork pulling the DNA apart? So there's really two major questions there, right? When you answer the question, make sure you address both of those questions. Okay, so let's draw that replication fork. Okay, so we draw the replication fork. So throw me off some enzymes here, or some proteins also. Helicase, yes. Helicase is going to break those hydrogen bonds. Okay? What's going to keep this thing apart when it has those hydrogen bonds apart? SSB, the single-stranded binding proteins of those proteins. We just draw them there, and we may draw them here and say, you know, SSB. Okay, what else? We have to put down, before we start transcription, before we start this, can DNA polymerase continue, can DNA polymerase start making DNA? Can it initiate that action? No. What has to be there first? A primer. What puts down a primer? Primase, RNA primase. Okay? So we might draw this and say, you know, RNA primase for the RNA primer. What's another thing here? What's going to make the DNA? DNA polymerase. Okay, so we'll draw some here, and we'll show maybe a blobby thing here and say DNA polymerase. Okay, good. So we've got DNA polymerase, and we've got our little short stretches here. Okay. Which is we have two strands. It's the what and what type of strands? The leading and lagging. Okay, this is the leading. This is the lagging. What else do we know? Or, well, yes, this all had to start at an oocyte. You're correct. But for this, you wouldn't have to worry about that. Because if you drew the whole, you know, chromosome here, the oocyte would be here, but that replication fork may be way down here. But you're correct. There would have to be some oocyte at some point, origin of replication. What are some other things we got here?
5' and 3'.
5' and 3', exactly. And then what links those, and it fills in the gaps. What's the last enzyme that fills that gap in? Ligase, because we're going to have that two bases come together. We want to fill that in with ligase. Okay? That's it. That would be sufficient for it. Okay, now, how does bacteria deal with excessive supercoiling stress during replication? What enzyme does it use? Gyrase, a type of topoisomerase. And what's it going to do? It's going to break one of the strands and allow the other strand to spool through it and then reconnect the strand. And that relieves the super stress. You know, and Cipro targets that action actually. So, okay. How is DNA methylation used in DNA replication and proofreading? How is it used? Initiation of replication. And how about the proofreading? How is it used in proofreading?
Protects against infections.
No, you're correct about how methylation will identify self-bacterial DNA. But how do we use it in proofreading? What is proofreading?
Making sure that the eggs
Correct. So let's just continue with this diagram. Let's just say we put our DNA down. This old strand has methyl groups here, here, and here, and here, and here. We have methyl groups here. As we're making our DNA, our template has these methyl groups on it. This does not. Okay? Now, what would happen if we saw, well, right here, because it's an exonuclease function, but let's just say we saw here A and a G. Does that work?
No.
Which is right?
Whichever one has the methyl.
Whichever one has the methyl group, because the methyl group is the template. So it uses methylation as essentially a tiebreaker. It says, okay, you have the methyl group. I'm going to believe you. G, you're wrong. Exonuclease activity. Pull that thing out, and it replaces it with what? A T. Exactly. That's why. Okay, and this DNA is called hemimethylated. Okay.
It's the one that has the methyl.
Well, if it has, one has methyl, one does not, it's hemimethylated. So the whole entirety of the structure is hemimethylated.
So are we supposed to put what you just drew in words if you say describe?
Yes. Or you know what? Okay. How this verbiage-wise? Well, how would I say it? I would say, or you could draw it and talk about that example. How would I say this as with words? I would say it uses RNA or uses methylation to identify which template strand, which strand is the template strand. Newly formed DNA does not have a methyl group. The template strand does have a methyl group. And then you could say, for example, this has an A, this has a G, this has a methyl group. It would select that one. All right.
Can we go over some of those fill in the blanks?
We sure can. Let's do that right now. Okay, so let's go with the first one. What is a covalent bond that connects two adjacent carbohydrates on a strand of DNA? Not hydrogen. Nope, that's a nope. No, you're right. You're right that we have two strands of DNA, but the two nucleotides, nucleotide 1, nucleotide 2, nucleotide 3 on the same strand. Phosphodiester bonds, that is correct. What's the central mass of DNA found in the center of a bacterium? Nucleoid, correct. Circular form of DNA found in many bacteria and mitochondria. This is not the chromosome. Not histone. Histone's a protein that the DNA will wrap around. Plasmid. Plasmid, correct. What makes short RNA primers complementary to the template DNA that gives DNA polymerase a place to begin?
Primates.
Primates, correct. This is the start of DNA replication. Okay, term used to describe one strand of mRNA that codes for two or more proteins. Polycistronic, correct. Also known as a ribosome binding site, location where on the mRNA the ribosome will bind to. Shine-Dalgarno, correct. Shine-Dalgarno. The next one we'll talk about for the next test, but that's a regulon. The next one, we're going to talk about for the next test, we're talking about operons. System uses that again, next test, that's going to be the SOS response system.
Which one's that one?
The system that uses DNA polymerase 4 and 5, SOS response. Or the SOS system. Are RNA and bacterial ribosomes used for phylogenetic analysis? We haven't talked about that, but 16S. And again, the other ones we're going to cover for later tests. So that's going to cover us for those tests. Okay, so let's look at those multiple choice. Okay, so the first one, don't worry about that one. The second one, we didn't cover that this time, but actually methionine is not the start amino acid. It's actually a kind of like methionine, but it's a little bit different. So anyhow, The third one, what is true about the genomes of bacteria? What is true about the genomes of bacteria?
1, 2.
Well, let's see. Typically, they have a singular linear. Well, no.
Bacteria is circular.
Circular. Haha. So 1 off of it. No. So 1 is incorrect. They do not have a nucleus around the DNA. True. By definition, they are prokaryotic. Okay. So, and DNA has a mix of introns and exons. False. So the only one true here is 2. So it's B. D. I'm sorry, my mistake. D. All bases in a codon are of equal importance. We didn't talk about that one. Well, we'll talk about that for the next test, but it is false. Have you ever heard of the wobble theorem? That if you look at a codon chart, the first and second are the important ones. The third can vary. So if you've had genetics, you might have probably heard of that one. The fifth one, that will be for the next test, but what does it break lactose down into? C. Galactose and glucose. Okay. Okay, how many phosphates are removed from should be form. I put form instead of from an incoming DNT to provide the energy to make DNA?
3.
No, 2. Correct. Has 3, but it removes 2. Okay.
We need to worry about the page before that. We're going to be on there.
I'm sorry?
Like 6 through 10, we're going to be on this tab.
Let me see. Hang on. Let me.
Just get these skipped.
I'm skipping the ones that will not be on this test.
Right.
Yes. But the earlier ones would. Let me go back and make sure about the. Okay, I see. 6, 7, 8, 9, 10. That's the ones. Yeah. Okay, 6. No, that's for the viruses. We talked about that. 7. That's the next test. 8. What's the functions of DNA polymerase 3? What is the functions of DNA polymerase 3? That's number 8. Well, does it add free DNTPs, such as the ATP, to an existing strand of DNA? Yep, sure does. Does it have exonuclease proofreading? No? Polymerase? Yeah? No. It's exo. It's exo. It has exonuclease activity. Hey, does it have exonuclease proofreading activity? Yes. And can it initiate DNA extension? No. It can continue DNA extension, but it can't initiate it. What does it have to initiate? Primase. A primer laid down by primase, yes. Okay. Okay, 9, we'll cover that with viruses. 10, we'll cover that for the next one. 10, for the full expression, if you're interested, is you have to have the presence of lactose and the absence of glucose. What's the typical size of a bacterial genome? 3 to 5 million bases, megabases, correct. And the functional group on carbon on the carbohydrate of a nucleotide, which prime number connects to the which prime number defines it being DNA or RNA? We didn't really cover this that much. This won't be on the test, but it's the 2' carbon. So what am I talking about with this? Okay, we have a little chimney on the house here. On the 1, 2, 3, 4, and 5. What's attached to the 1' carbon? Our not methyl hour. Our base, right? A nitrogenous base, be it A, T, G, or C. 1'. What's on the 2'? Well, this, let's say the word DNA and RNA. This is not on the test, so we didn't cover this, but this is more of an FYI. DNA, RNA. What does DNA stand for?
Yeah.
Deoxyribonucleic acid. So deoxy. D Latin being from or away from. So this has oxygen away from it. Deoxyribo. Okay, so we're missing an oxygen relative to the ribose. So in DNA, we would have a hydrogen here. And RNA would have, guess what? An OH, would have an oxygen. And that's the major difference. Okay? That's what defines it as DNA or RNA. Oxygen, no oxygen. Deoxy, an oxygen away from. Okay. 3' carbon, that's the end where the phosphate from the next one will attach to. 4', 5' carbon will be where the other phosphate attaches to. Okay, we didn't cover that for this test, but just an FYI on this one. All right. Okay, so that pretty much covers this test. I thought I had all other old tests up there. I'll get all the other old tests up. Okay, I will get those up for you because I've taught this class last semester, and I know the tests are out there. I return them, and it's not fair that other people have the old test and you guys don't. This is not fair. So I will get those to you. So any questions now? That wraps up what would be on the test. Expect a similar format test. Okay? Shoot.
About the cell wall, the alternating pattern of MGs.
Right.
Do we need to just know that it alternates or actually draw that?
Well, I'll tell you, it's the cell wall that's really important, and I would be able to draw it, to be honest. Okay. Because it's important for a lot of reasons. One, it's just one of those bullet points with microbiology. You have to know about the cell wall. And then as you get into microbiology, you start talking about how antibiotics work, for example. That becomes really important. Like when you start talking about how vancomycin works, how penicillin works, things like this, that becomes a really important point. So be able to at least draw that too. I would suggest being able to draw it.
If we cover 2.5 test words, is our test going to be longer, or is it going to be about the same length as one of these?
Well, the thing is, it's going to be about the same length as this. What we've done is I've kind of rearranged how I went over things to make it better for the summer. There's some things that were, say, on this test that will be on another test for us. And there's some things that we have covered that is not on this test. So when I upload those other tests, you're going to see that there's some things from the other test that we've covered. For the most part, though, a lot of the material is on this test. But I've rearranged the material to make it better for summer.
And the historical stuff, it was on your slides, but we didn't cover it.
Maybe don't worry about it then.
Okay.
Oh, man.
Oh, the typhoid. Okay, the historical things like Pasteur.
No, yes, that's.
Okay, Pasteur, all that. No, if I didn't mention, I left that out because I was. The pathogen, yeah, you're going to expect some questions about those. So let me ask you a few questions about that one. What bacterium causes Lyme disease? Not bacillus, Lyme disease. What bacterium causes Lyme disease? Burgdorferi, Bordella burgdorferi, named after old Willie. He's still actually alive, actually. Burgdorferi, but yeah, it's Borrelia burgdorferi. Okay.
But do we need to know like symptoms and treatment, or do we just need to know like the names and
in Health Micro, you need to know a lot more about it than you do in here. Let me say it that way. So I'm not going to get down so far down to like you're going to give this antibiotic for treatment. That's not what I would ask. But I would be more general. Okay, so I would be a lot more general in it, and if I ask questions, more of them are going to come from like the connection to microbiology. For example, Bordella burgdorferi. What does it have that allows it to move? What type of flagella? Endoflagella. Because we covered the flagella a lot more. So I would ask more questions like that. Okay. I might give some major things about symptoms, but it'd be a major one. Like, what's the major symptom of a Lyme disease infection? Bullseye rash, right? But I wouldn't get so specific as to say, what would you treat it with? Which antibiotic? I would not get that specific. Okay. Or another example, syphilis rates. We talked about syphilis rates. Would I say what's the syphilis rate per 100,000 in Alabama? No. What I say is compared to something like gonorrhea, is syphilis rates high or low? Lower. They're pretty low. Okay. What bacterium cause of diarrhea in hospitals? C. diff, Clostridium difficile. Bacterium that can cause extreme muscle contraction. Tetani. What two genera of bacteria can sporulate? Clostridium bacillus. Bacterium found in food, and as it as it grows, it releases a toxin that can cause muscle relaxation, used in medical situations to treat wrinkles and botulisms. But what bacterium? Correct. Okay. Bacterium that we obtain for GMOs, genetically modified foods. We'll obtain DNA from these, and we'll put them in plants. You've probably had some of these proteins if you've had any kind of corn products. You probably have these going around you right now. What bacterium do we get this from? Yeah, I worked with. So you see, okay. So I worked with BT a lot back in the day. We call it BT for a reason. Bacillus thuringiensis. Thuringiensis. So what's the origin of this word? Okay, Genesis, or like Genesis, the original one. It was first identified in insects from a mountain range in Germany, Thuringia. All right, not a mountain range, a province in Germany. So it originated from Thuringia, Thuringiensis. What do you know? Okay, so Bacillus thuringiensis. Yeah, try spelling that in a thesis 80, 90 times. Anyhow, thank God for macros, because I had to cap THU, and every time I did THU, it filled in thuringiensis for me. So, thank you, macros. Okay. Okay, so let me ask you a few questions here while we're thinking about it. What helps us initiate transcription? RNA polymerase, but what initiates that RNA polymerase? What do we have to have? RNA polymerase coming on there. Can any RNA polymerase just sit on there? What has to be there? Not a primer. RNA polymerase. We're talking about transcription translation. What factor was necessary? Sigma factor. Okay. And again, that's how bacteria are going to regulate a lot of their things. Some sigma factors are going to be there, some sigma factors are not. Okay. How about sporulation? Can you tell me the steps of sporulation? What are the steps of sporulation? Well, first of all, someone said Bacillus and Clostridium do this, but what are the steps of sporulation? What are the steps? You tell me. What are the steps of sporulation? So we got a bacterium, right? I believe this was a essay prompt on a previous test. So I will get those tests up to you today. What are the steps in sporulation? Step 1. We have a bacterium, and what does it have in it? DNA, right? Okay, so we got our DNA in this bacterium. And what do we kind of liken sporulation to be like? It's like cell division without the division. Okay, so what's it going to do? It's going to replicate this DNA, attach it to the side of the wall here, and it will, the cytoplasm here, and it will duplicate its DNA, and as it duplicates its DNA, what happens? We have 2 distinct regions of DNA in it. So we've got 2 copies of DNA in it. And what's the next step? So we're going to start forming what we would call a septum, a wall between the 2. And this was defined as the what? The mother. And we call this the what? Bless you. Forespore. At some point what we call the forespore as it develops. And what are we going to start laying down? We're going to lay down those cortex proteins, and eventually we're going to do what? We're going to form a mother cell, this DNA, and here we're going to form an environmentally resistant endospore that has the same DNA in it. Then what's next?
It's released from the original cell.
Correct. This is released from the original cell. So this endospore releases into the environment. So the spore here is released. The spore has the DNA in it. Okay. The spore then finds an appropriate area of nutrients 5, 10, 20 years later, whenever. What's it going to do? Germinate. Correct. Now, in a lot of microbiology books, there's multiple steps, like step 1, step 2, step 3, step 4, step 5 for sporulation. If you have one of those microbiology books, don't memorize every specific seminal step. Okay? Just the major points of sporulation. What's in the PowerPoint? Okay? But, you know, technically there are well-defined, like step 1, step 2, step 3. Okay. Alrighty. Let's see what else we got here. Let's talk about taxis. Let's talk about taxis. How does a bacterium move? Flagella, right? And what's the ratio? What's the idea of runs and tumbles? This was another question prompt on a previous test. What's the idea of runs and tumbles?
Runs in the same direction and tumbles.
Is randomized movement. Exactly. Now, if you're going towards something you want to, if you're going the direction you want to, do you have more runs or more tumbles? More runs. If you're going towards something you don't want to go toward, what is the ratio? More tumbles, because you're thinking that the random motion of a tumble will, you know, at some point, point you in the right direction. Okay. We talked about the different flagellar patterns. Expect a couple questions on those. 1 or 2 at least on those. What are the 3 points to a bacterial flagella?
Is that the
Hook, you're correct. What else?
basal body?
Basal body and filament. Where do we add our flagellin protein on the filament? On the top, on the end of it. Okay. And movement of what causes the flagella to move?
The basal body.
Basal body. And what specific thing moves with the basal body to cause motion? Not water.
Motor.
The motor. What causes the motor to spin? Not water. No. Protons. We're going to move protons through this. Or in some cases sodium, but sodium or protons will work for that answer. Okay, we talked about cell layers surrounding a bacterium. What are the two major types of cell layers surrounding a bacterium?
The membrane and
Not the membrane. We'll get to that pretty soon, but the exterior. We have the cell wall, we have the bacterium, but sometimes we have an exterior layer around it. What are those two layers? Slime layer and the capsule. If you had to give me a basic thumbnail sketch of those, and what are they made of? What polysaccharide? What polysaccharide makes up those layers primarily?
Is it lipo?
Not lipo. No. No. You're close. Lipo. It sounds like lipo, but with a G.
Glyco?
Glyco. Yes. Yes. Glyco. What? Glycocalyx. Glycocalyx. Polysaccharide. So we got the slime layer and we got the capsule. Now the slime layer, as the name implies, I'm going to guess most of you are too young for Ghostbusters. Remember Ghostbusters? Okay, and then remember the cartoons too? They had the Ghostbusters. Okay, anyhow, had Slimer. It's all kind of like gooey and ooey. Okay, slime layer is more of a gooey, loose packed layer. Okay, capsule is a more dense layer. Okay, the capsule is more firmly attached to the bacterium. Slime layer is more loose attachment. Okay. Okay, but glycocalyx, as the name implies, mostly polysaccharides, have some proteins in it also. All right. And what's that, when all the bacteria get together and they start extruding all this glycocalyx and they make a big packed thing of bacteria, what do we call that? A big, just like super grouping of bacteria cemented together with its glycocalyx. Really important, when you start talking about microbial ecology, these things are huge in that area. Also, you see this in medical situations. These can form on medical implants. It's a major problem. Causes a lot of times we have to go and replace transplants because of this. Kodak used to make this, and you'd put this in cameras. Film. Biofilm.
And that's a group of bacteria.
It's a group of bacteria who has extruded this glycocalyx, and essentially they have packed themselves into a sheet of multiple bacteria. And there's a lot of reasons for this. There's, protection in numbers. It's going to protect them. They can adhere to, think about a stream, right? You can adhere these colonies of bacteria to that stream. Actually, there's even some communication in these biofilms between the bacteria. So it's a really interesting topic, but it's more for microbial ecology. When you take a class like that, you really do talk about things like biofilms. Here, we just mentioned what they are. Okay.
We need to know all the different shapes and arrangements.
Yes. Yes, yes, yes. Absolutely, positively, yes. Yes.
Shapes and arrangements of what?
Shell shapes and cell arrangement. So we define bacteria, we can define them in two ways, the arrangement and the shape. So if it's round, what is that? Toxide. If it's rod-shaped, it's bacilli. If it's comma-shaped, it's not, no. If it's comma-shaped, Vibrio. Vibrio. Spiral-shaped, it's Sporula, okay. Oh, and then, okay, now, if you have a clumping of them, what is that? Staph. If you have a long strip of them, it's strep. Remember strip and strep rhyme. Okay. I'll have to say, there's a lot of material in this test, isn't there? That's the summer. That is the summer. Okay, these are things inside of a bacterium that are kind of like rainy day things, that we pack stuff in a bacterium that we can use later on. Inclusions are inclusion bodies. It's like, you know, we pack our fat right here. Well, bacteria are going to pack their fat in a specific area, inclusion bodies. Okay.
Are any vocabulary words that you have throughout the PowerPoint going to be really important too?
Yeah.
Like you have incubation and you have the certain words that you have.
Yes. Yeah, those would be, you should know those words, yeah. Yeah. So incubation, how about when you add a bacterium to a particular growth medium? That's what? Not incubation. Incubation is keeping it at a specific temperature and inoculation, correct. Okay, and bacterial food we call media or medium. Medium is a singular, media is the plural, if you want to get all technical about it. How about a bacterium that grows between, you know, about 25 to 40 Celsius? What do we call those? That's another topic we talked about. Mesophiles, correct. What a bacterium that can survive at elevated temperatures for a short period of time? Thermoduric. Okay. Okay, so we talked about the temperature thing too. We talked about growth relative to oxygen availability, didn't we? So tell me some ones about that. Let's say I have a picture of a tube here. And I want to say the depth of this tube is relative to the amount of oxygen. I have a lot of growth at the top. What do I have? Aerobe. I have no growth at the top, but a lot of growth at the bottom. What do I have? Anaerobe. If I have some growth, not exactly at the top, but a little bit below, what do I have? Microaerophile. What is a pathogen that we see in medicine that causes ulcers that survives in lower oxygen environments? Helicobacter pylori. Classical example. It's going to survive at lower rates of oxygen availability. Too high, it's not going to grow very well. Too low, same thing. It needs a lower amount of oxygen. Find that in the stomach. Major cause of ulcers. Okay.
And that was what?
The bacterium?
Yes.
Helicobacter pylori. And remember, that was the scientists that did that, that tested it on themselves, and they actually got the Nobel Prize in medicine a few years ago for that. So, if I remember right, they were an Australian team. Okay. So we got that, and then what if we have bacteria that's growing all throughout this? Those are aerotolerant. Okay, so that's, how could I have a question like that? I could say, here's a tube right here, and here's growth just right here. This is an example of what? And you'd have to say, you'd have to fill the word in. Or it could be a multiple choice. Or it could be this bacterium grows in less than normal oxygen environments. For example, Helicobacter pylori.
So there won't be any pictures on this where we have to label?
There could be. There could be. There could be pictures. There definitely could be pictures. I could have something like this, you know, a series of tubes on an essay question, and say, consider this picture below. Show me how the microaerophile would grow. Show me how these would grow. And again, my essay questions, I'm going to give you a few prompts, and you pick the ones you want. So,
would you, okay, in some of your pictures that are in your slideshow, would would you put something on there and be like, and circle something and be like, tell me what this is? Would you do stuff like that?
I've not done that for a test last semester in micro, but no, actually, I take that back. I did do that for a test. I did, but only one time. So would I possibly do that? Yes, but it would be something major. It would not be something minor. Okay, let me. And what do I mean by that? Let me find a picture here and show you what I mean. For example, I might have a picture like this, and I would obfuscate all the words, and I'd point to that pink thing, and I would say, consider this, what type of flagella is this? Which is endoflagella. Okay, I'm not going to be super, it would have to be a major thing. Okay. Okay, so let's see what else we got here. We have our definitely know how to draw the cell wall. Definitely know how to draw the cell wall. Expect definitely a question on that. What's the space? What's the space between the cell wall and the cell membranes and gram-negatives? Got a special name for that. Periplasm, correct. And on that LPS layer on the outside, we have something. What do we have on that layer? Let me say it this way: what's an LPS? Lipopolysaccharide. And why are these medically important? And where do we find these LPSs? Gram-negatives. So lipopolysaccharide, what does that imply? Fat, there's a lipid, and a sugar. Exactly, we've got a long string of sugars. What are these LPSs implicated in? The release of LPS typically causes what? Shock, yeah, drop in low blood pressure. As we break apart these gram-negatives, they release these LPSs, and the endotoxin on the end will actually induce a shock effect. So
That's in gram-negative?
That is in gram-negative, correct.
You said the periplasm is also gram-negative.
Correct.
When you say that we have to know how to draw it, we don't have to like go in depth or anything. Just show the.
Okay, so let me draw how I would draw it, and I'll show you. And again, people ask the question, how would I grade test questions? And I'll show you. Back in my day, there was these books. It was Johnny Do Right or something like this, and the book was like, "This is how Johnny does right." And it's like he puts his pocket knife away, and this is how Johnny does wrong. He has a pocket knife and he's waving it out like this. So I'm going to show you how I've seen people answer these questions and they don't get full credit. Then I'm going to show you how people do answer these questions and they get full credit. Okay, so I say, draw me the gram-positive cell wall. No, this is what you don't want to do. Gram-negative. And I have seen things like that. What the heck does that tell me? Nothing. Don't do that. How do I want you to see you draw this? How's a yes to this? How's a good way? Well, let's start talking about the gram-positive. And if you look on the prompt, you're going to see I want to know where the outside and the inside part is. So I have a cell wall. I might have some teichoic acid. I might have some lipoteichoic acid. Okay. The cell wall, I would say, made of peptidoglycan. Okay. What comes under the cell wall? The cell membrane.
In the diagram in your slides, it looks like there's a space. Is there a space in between those as well?
That's actually a debated point. It would possibly, but is a function like the periplasmic space? We don't know. Okay, and then this is the cell membrane. You might, if you want to be high speed about it, you might even draw a bacterium here, like a full bacterium, and show me like a cross-section type of thing. Seen that. Then you'd want to say this is the outside, and this is the inside. Okay? That would be a good response. Gram-negative. Gram-positive is going to be pretty straightforward, you know. Okay, gram-positive is pretty straightforward. Gram-negative is where you're going to make it break it. So, you have, on the outside, you have what layer? You have those LPSs, right? You have those carbohydrates, and on the ends of those carbohydrates, you have the what? Fatty part? Those fatty parts are what lodge in the cell membrane. Okay. And that cell membrane, what's next? Right, we have the cell wall. And is that cell wall next to the cell membrane? No, it's separate. And then what's next?
The bilayer.
The next bilayer, correct. Back in my day, I'm not saying to do this, but back in my day when I do this, I was legendary for this apparently. I had a thing of colored pens and pencils. And I'd come in with my little bag, and I would draw this, and I would draw all the hydrophobic, I would draw with red lines. And all the hydrophilic, I would draw with blue lines, or blue circles. And I would have like a color code chart on the side. I'm not saying to do that, but that's how I would do it, and have like a little legend over here, and would have like what each color meant. Yeah. Okay. So, we have a cell wall right here, right? And here's our inner membrane. And here is our outer membrane. Here is our LPS. And what's this space called? Periplasmic space. All right, and this is not shown really well here, but this is a bilayer also. Bilayer space with the cell wall and another bilayer. Okay, do you see how that's different than the last picture? I have seen pictures like that, trust me. I might give them a point or two. But make sure you have everything like that. Okay. Okay, another question on another test was another question here. What pathogen is implicated in elephantitis? Gets in the lymphatic system. The very first one. That you're right, but that's not the right answer. But yes, Wucheria bancrofti. Wucheria bancrofti gets in the lymphatic system, clogs it up. One more here, and then well, I'll go as long as you guys want to. But it looks like people want to cut bait here. Differences between bacteria and archaeal cell membranes. What are some differences? No, that's not one of them. They're about the same. They're about the same. let's talk about what makes them up. What makes up bacterial cell membranes?
Phosphate group.
Phosphate groups, and then what are the hydrocarbons made of? Fatty acids. What do we see in archaeans or archaea? Repeating isoprenes. Isoprenes is a carbon-carbon chemical group. You don't need to know the actual structure of an isoprene. It's a carbon group like this with a double bond in the middle. It's just a functional group in chemistry. It's a 4-carbon. No, it's a 3-carbon. In bacteria and us, it's not that. It's just a simple carbon-hydrogen thing like that. Like that. So it's a little bit different structure. There's methyl group sticking up. There's no methyl group sticking up on this structure. With an isoprene, you have a methyl group sticking up every carbon going down. Okay.
Do the archaeal cell membranes have the hydrophilic and hydrophobic?
They do have those. Yes, that's another difference, though. Archaeal cell membranes do have hydrophilic, hydrophobic. But with bacterial, the hydrophilic is going to be what? As a phosphate group. That archaeal, sometimes phosphate, sometimes other hydrophilic groups. What's another difference? How about mono versus bi?
Mono and archaeal.
Right. Archaeal can have mono or bilayers, and bacteria have bilayers. We have bilayers. But archaeal, especially the extreme thermophiles, they're going to have a monolayer. Another difference? Different glycerol linkages. Ester in bacteria in us, ether in archaeal cells. A little bit stronger of a linkage. What's another difference? One more difference and we'll be done. How about the presence of ring structures in it? If you look at an archaeal membrane, sometimes there's actually rings that form on the hydrocarbon part. Ester and ether, that's a different thing. The ester and ether is what connects the hydrocarbon chain to that polar group. So, all right, any more questions? Yes, ma'am.
Are you going to lecture after the exam?
Unfortunately, yes. I mean, I don't want to. Honestly, I don't want to, but the how summer works is I have to. So I know you guys don't want to hear me do it, and I know that, but it's just the schedule. It has to be done. So it is what it is.
Can I ask you a question that has to do with the test?
You bet.
When's our lab practicum?
Probably the middle of next week. So.
What's in the lab kit that we have to spend money on?
Well, the thing is, you buy the lab. We'll have a lab kit check next week too. Is we make you buy the kit, and then we get some of that money back, and then we'll use the media and everything else to pay for it. So it's not like you have to. It's not like, well, I got these pieces of paper and a pen. I can go to Walmart for $1 and buy this. It's money built into the kit, so we can buy supplies for the lab. Because we run, we're going to run through a lot of media when we're doing the unknowns. You're going to go probably through 30 or 40 plates at least. And it just helps us get money for that.
Are you going to sell this game?
I am. It'll be 50 cents a pop. All right. Any more questions? Good review? Okay. Then if you have any questions, email me. I'm pretty good about getting with email. Some of you have emailed me before, I think, and I'm fairly fascinating back to you. So let me know if I have any help, and good luck.